This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to this exploration of one of nature's most captivating features, the plant petal. Today, we'll uncover the remarkable science behind these delicate structures. From their evolutionary origins to the chemistry that creates their stunning colors, we'll discover how petals attract pollinators, regulate temperature, and even communicate. You'll learn why some petals shimmer in ultraviolet light, invisible to our eyes, and how these seemingly fragile structures are engineering marvels. Join me as we peel back the layers of botanical beauty to reveal the fascinating science within. Chapter 1. The Chemistry of Color. When we admire the vibrant crimson of a rose or the sunny yellow of a buttercup, we're witnessing the culmination of intricate molecular processes that have evolved over millions of years. The colors adorning flower petals aren't merely decorative accidents of nature. They represent sophisticated chemical and physical systems that plants have developed to communicate with the animal world, protect their reproductive structures, and regulate their own physiology in response to environmental stress. The science of petal coloration divides into two fundamentally different mechanisms. The first involves biological pigments, specialized molecules embedded within petal cells that selectively absorb certain wavelengths of light while reflecting others. The second mechanism, structural coloration, depends not on chemistry but on physics, using microscopic surface features to manipulate light through interference and diffraction. Many flowers combine both strategies, layering pigments and structures to create colors and visual effects that single mechanisms alone could never achieve. Let's begin with anthocyanins, the pigment family responsible for most of the reds, purples, and blues we see in the plant kingdom. The name itself comes from the Greek words for flower and blue. Though somewhat ironically, these molecules produce a much broader spectrum than their etymology suggests. At the molecular level, anthocyanins are built from ring-shaped structures featuring alternating single and double chemical bonds. This alternating pattern creates what chemists call a conjugated system, where electrons can move freely across multiple atoms. These delocalized electrons make anthocyanin molecules highly reactive to photons, the fundamental particles of light. When a photon strikes an anthocyanin molecule, electrons absorb the energy and jump to higher energy states. The wavelengths that get absorbed depend on the precise molecular structure, while the wavelengths that aren't absorbed bounce back to our eyes or to a bee's compound eyes, creating the perception of color. A red geranium appears red because its anthocyanins absorb green and blue light, reflecting primarily red wavelengths. Change the structure slightly, and you get the deep purple of a violet or the brilliant blue of a delphinium. What makes anthocyanins particularly fascinating from a scientific standpoint is their remarkable instability. These are temperamental molecules, easily influenced by their chemical environment in ways that dramatically alter their color output. Temperature fluctuations can shift their hue. The pH of the cellular fluid surrounding them acts like a chemical switch, transforming reds into blues or vice versa. The presence of metal ions like iron or magnesium creates yet another layer of complexity. Even interactions with other molecules in the cell can modify anthocyanin behavior. Plants have developed sophisticated strategies to stabilize these volatile pigments and maintain consistent colors. Many flowers sequester their anthocyanins inside specialized cellular compartments called vacuoles, where the pH can be precisely controlled. This is why a hydrangea growing in acidic soil produces blue flowers, while the same genetic variety in alkaline soil blooms pink. The plant isn't producing different pigments, but experiencing the same anthocyanin molecules in different chemical contexts.
Some flowers go further, building elaborate molecular scaffolds to hold their anthocyanins in place. They stack anthocyanins together with other flavonoid molecules, creating layered structures reinforced with metal ions. These stacks provide mechanical stability while also fine-tuning the exact wavelengths of light the pigments absorb and reflect. The difficulty of maintaining stable anthocyanin systems may help explain a curious gap in the floral color palette. True blue flowers remain relatively rare in nature compared to reds, purples, and other hues. Creating and maintaining the precise chemical conditions needed for blue anthocyanins requires such specific cellular chemistry that many plant lineages simply haven't evolved the necessary machinery. This same challenge has frustrated horticulturists for generations in their attempts to breed blue roses and blue carnations. The chemical sensitivity of anthocyanins does offer plants certain advantages beyond color production. By tweaking the number of hydroxyl groups attached to the anthocyanin ring structure, plants can shift colors across the spectrum from red toward blue. This molecular flexibility allows different populations of the same species to produce distinct flower colors, potentially attracting different pollinators or adapting to different environmental conditions. This instability allows some flowers to shift colors dramatically in response to pH changes or other triggers within hours or days.
While anthocyanins dominate the cool end of the color spectrum, carotenoids paint petals in warm yellows, oranges, and reds. The marigold's golden petals owe their color almost entirely to carotenoid pigments, as do the cheerful yellow faces of buttercups, the vibrant orange of California poppies, and the soft cream of daffodils. Structurally, carotenoids look quite different from anthocyanins. Rather than rings, they're built from long chains of carbon atoms connected by alternating single and double bonds. This linear architecture, with its conjugated system, creates a different pattern of electron behavior. Just as with anthocyanins, the alternating bonds allow electrons to become delocalized across the entire molecule, making carotenoids responsive to light energy. But the chain-like structure means that molecular length becomes a critical variable. Scientists have identified over 700 naturally occurring carotenoid variants, and the length of the carbon chain directly determines which wavelengths of light each molecule can absorb. Longer chains absorb longer wavelengths. Molecules generally need at least 8 alternating double and single bonds to produce visible color. Beta-carotene, one of the most common carotenoids in both flowers and foods, contains 11 such bonds. The sunflower's yellow petals contain high concentrations of carotenoids, as do the petals of the yellow monkey flower. When you bite into a carrot or slice open a mango, you're eating beta-carotene, which your digestive system breaks in half to form 2 molecules of vitamin A. Your body then converts that vitamin A into retinol, a molecule essential for vision. There's a beautiful symmetry here. The same class of pigment molecules that allows plants to create colored signals that we can see also provides the raw material for the biochemical machinery that lets us perceive those colors in the first place. Carotenoids tend to be more chemically stable than anthocyanins, less prone to shifting their colors in response to environmental fluctuations. This reliability makes them particularly useful for flowers that need to maintain consistent appearance across varying conditions. However, plants can still manipulate carotenoid-based colors by adjusting the concentration of pigments, the specific carotenoid variants they produce, and how they layer different carotenoids together in the same cells. Some carotenoids, called xanthophylls, reflect not only yellow wavelengths visible to humans, but also deep violet in the ultraviolet range that bees can perceive but we cannot, creating communication channels invisible to our eyes. The scarlet monkey flower demonstrates how combining anthocyanins and carotenoids allows for colors neither pigment could create alone. By varying the relative concentrations of both pigment types, different populations or species can generate a remarkable range of hues from the same basic molecular toolkit. A flower that looks predominantly yellow might have high carotenoid levels with minimal anthocyanins, while a deep orange bloom could represent a balanced mixture, and red flowers might layer anthocyanins over a carotenoid base. Beyond visible coloration, certain anthocyanins and flavonoid compounds absorb ultraviolet wavelengths shorter than about 400 nanometers, while appearing pale in visible light to human eyes. These UV-absorbing patterns remain invisible to us, but create striking bullseyes and radial guides for bees and other insect pollinators, whose visual systems detect ultraviolet radiation. By absorbing ultraviolet radiation, these flavonoids protect pollen grains from mutations and degradation that UV exposure would otherwise cause, serving both signaling and protective functions simultaneously.
Beyond pigmentation, the shape of petal epidermal cells also affects appearance. In 75 to 80% of angiosperm species, these cells develop a distinctive conical or papillate shape, projecting outward from the petal surface. These cone-shaped cells create microscopic textures that serve multiple functions. They focus light to enhance color saturation, create the velvety appearance of many petals, affect overall petal shape, decrease surface wettability, and provide mechanical grip for visiting pollinators. The distribution of conical versus flat cells across the petal surface can itself create pattern elements that contribute to visual complexity. Petal surfaces can also develop specialized structures called trichomes, hair-like cellular extensions that serve diverse purposes. Cotton flowers produce trichomes that physically entangle adjacent petals together, ensuring proper unfolding as the bud opens. Snapdragons develop glandular trichomes inside the corolla tube that produce scent compounds while simultaneously trapping pollen grains, ensuring they contact visiting insects rather than falling to the ground. But pigments and surface structures represent only part of the story. Some of the most spectacular colors in the plant kingdom arise not from colored molecules at all, but from the physical architecture of the petal surface itself. This is structural coloration, a phenomenon that works through the interference of light waves rather than their selective absorption.
The principles underlying structural color in flowers mirror those that create the shimmering blues of morpho butterfly wings or the iridescent greens of peacock feathers. When light waves encounter tiny structures comparable in size to their wavelength, those waves can interfere with each other. If surface ridges or layers are spaced just right, certain wavelengths will reinforce each other while others cancel out, creating intense, pure colors that seem to glow from within. Hibiscus trianum demonstrates this principle, with nanoscale ridges on proximal petal cells creating an iridescent blue shimmer visible to pollinators. These ridges, measuring just hundreds of nanometers across and arranged in parallel arrays, act as diffraction gratings that split white light into its component colors. The specific spacing and height preferentially reinforce blue wavelengths while scattering others. What makes this system even more intriguing is how these nanostructures form during petal development. The ridges aren't built through a carefully orchestrated construction process where the plant deliberately sculpts the pattern. Instead, they emerge through a more elegant mechanism involving mechanical forces. As petal cells grow and expand, the outer cell wall grows at a different rate than the cuticle layer covering it. This differential growth creates compressive stress, and when that stress exceeds a critical threshold, the surface buckles, forming parallel ridges. It's the same principle that causes a piece of paper to wrinkle when you push its edges together. This buckling mechanism helps explain why iridescence has evolved repeatedly across flowering plants. Any flower could potentially develop these nanoridges simply through modifications to how cell walls and cuticles grow relative to each other. The phenomenon represents convergent evolution driven by physics rather than requiring complex new genetic machinery. Tulips demonstrate another form of structural coloration through different mechanisms. Some tulip varieties produce iridescent sheens across their petals, with colors that shift as you change your viewing angle. These effects don't come from pigments, but from microscopic surface structures that manipulate light through interference. The structures are subtle enough that they don't overwhelm the underlying pigment colors, but add an extra dimension of visual complexity that helps the flowers stand out. Edelweiss, the iconic mountain flower, employs photonic crystals in its white bracts.
These aren't crystals in the gemstone sense, but rather highly ordered microscopic structures with regularly repeating patterns that interact with light waves. The spacing and arrangement of these structures determine which wavelengths get reflected and which pass through, creating the flower's distinctive white appearance enhanced by subtle light-scattering effects.
The distinction between pigment-based and structure-based coloration has practical implications beyond aesthetics. Structural colors tend to be more resistant to fading than pigment colors because they don't depend on maintaining specific molecules that can degrade over time. A flower could lose all its pigments and still retain structural coloration as long as the physical architecture remains intact. However, structural colors are typically visible only from certain angles and under particular lighting conditions, which is why flowers often combine both strategies. When plants layer structural coloration over pigment-based colors, they create effects that neither system alone could achieve. The combination can produce more intense colors, wider viewing angles for iridescent effects, or subtle variations in hue that change as a bee or butterfly moves around the flower. These layered systems provide richer information channels for communication between plant and pollinator. The buttercup illustrates this layering beautifully. Its petals appear uniformly yellow to human eyes, colored by carotenoid pigments. But high-resolution imaging reveals that the petal surface also features structural elements that enhance the color's brightness and create a glossy sheen. The structural features focus and intensify the reflected yellow light, making the flower more visible at a distance or in dappled forest light. These layered systems provide richer information channels for communication between plant and pollinator. Understanding how plants control and modify these different coloration systems opens windows into both development and evolution. A plant can alter its color by changing pigment chemistry, adjusting pigment concentration, modifying the pH or metal ion content of its cellular environment, changing the architecture of surface structures, or combining these approaches in various proportions. Each strategy offers different advantages and trade-offs in terms of metabolic cost, stability, visibility to pollinators, and protection against environmental stressors like ultraviolet radiation. The convergence of chemistry and physics in petal coloration raises questions about how these systems first evolved and how they continue to diversify. When flowers change their colors across evolutionary time, which aspects tend to evolve most rapidly, and which remain conserved? These questions lead naturally toward considering not just how individual petals achieve their colors, but how those colors organize into complex spatial patterns across the petal surface, creating the nectar guides, bullseyes, and intricate markings that make each flower species recognizable and that guide pollinators toward their rewards. Chapter 2. Invisible Boundaries. A petal uniformly colored in vibrant yellow or deep purple represents an impressive feat of biochemistry and physics, but nature rarely settles for uniformity when complexity offers advantages. Across the flowering plant world, petals display intricate spatial patterns where regions of contrasting color, texture, and cellular architecture create maps that guide pollinators to their rewards. A violet's delicate streaks pointing toward the flower's center, a sunflower's dark disk surrounded by bright rays, or a hibiscus's burgundy bullseye against white petals all represent solutions to a fundamental challenge: how to direct a visiting insect or bird to exactly the right location for efficient pollen transfer. These patterns raise questions that uniform coloration never poses. When cells across a petal surface all originate from the same undifferentiated tissue, what causes some to develop dark pigmentation while their immediate neighbors remain pale? How does a flower establish sharp boundaries between regions? And once established, how are these boundaries maintained as the petal grows from a tiny primordium measuring fractions of a millimeter to a mature structure hundreds of times larger? Why do closely related populations of the same species sometimes produce dramatically different pattern proportions? And do these variations matter to the insects and birds that visit them? The violet provides an accessible entry point into these questions. When you examine a violet flower closely, you notice delicate lines radiating from the flower's center outward across the lower petals. These markings, often darker than the surrounding petal tissue, create what botanists call nectar guides or honey tides. The name describes their function with admirable directness. Bees landing on the petal follow these visual pathways toward the center of the flower where nectar awaits. The guides act like runway markings at an airport, directing traffic toward a specific destination. The cellular basis of these patterns involves precise spatial control over pigment production. In the cells that form the dark streaks, the anthocyanin biosynthetic pathways detailed in Chapter 1 operate at high concentrations. In the cells between the streaks, those same pathways remain largely inactive or produce pigments at levels too low to create visible color. The resulting contrast makes the pattern conspicuous even on petals already colored by background pigmentation. As Chapter 1 discussed, many patterns extend into ultraviolet wavelengths that pollinators, but not humans, can perceive, creating additional guidance systems invisible to our eyes. The distribution of different cell types across the petal surface can itself create pattern elements. As explored in the previous chapter, conical epidermal cells serve multiple functions, including enhancing color perception and providing pollinator grip. In some species, the distribution of conical versus flat cells across different petal regions creates additional visual complexity beyond pigmentation patterns alone. Nigella damascena, commonly known as love in a mist, demonstrates how elaborate these cellular patterns can become. A single petal in this species contains at least 10 distinct subdomains, each characterized by different cell types and specialized functions. Some regions feature conical cells that enhance visual signals. Adjacent areas display flat pavement cells, creating different optical properties. Certain zones contain secretory cells, forming nectaries that reward pollinators. Other regions develop polygonal cells, short trichomes, or long hair-like structures. Each cellular domain must be specified during development, with boundaries established and maintained as the petal grows. Understanding how these patterns form during development requires asking a fundamental question. When does a growing organ specify regional differences that will eventually create distinct functional domains? The flower of an hour, Hibiscus trionum, has emerged as a powerful system for investigating this question. Mature flowers of this species display a conspicuous bullseye on the adaxial surface of each petal. The proximal third of the petal appears purple to burgundy, with cells that are flat, elongated, heavily pigmented with anthocyanins, and covered by a striated cuticle, creating iridescent blue effects. The distal two-thirds of the petal contrasts sharply, appearing white with conical cells and smooth cuticles. The boundary between these regions forms a remarkably sharp line, consistently positioned approximately one-third of the distance from the petal base to the tip in wild-type individuals. Researchers at the John Innes Center studying hibiscus petal development wanted to know when this boundary becomes established. Does it appear only late in development when pigmentation makes it visible, or does something earlier in the growth process already mark these future domains as different? To answer this question, they developed an imaging pipeline that captures petal morphogenesis at cellular resolution across multiple developmental stages. The technical challenges are substantial. Hibiscus petals develop inside floral buds with the adaxial epidermis, the future upper surface, facing inward and tightly packed against other floral organs. Observing cells on this inner surface requires carefully dissecting petals from buds at precise developmental stages, staining the cell membranes to make them visible under confocal microscopy, and then using software such as morphographs to segment individual cells and track their behavior over time. When researchers applied this approach to hibiscus flowers, they discovered something remarkable. The bullseye boundary exists long before any visible sign of pigmentation appears.
By measuring cell sizes, shapes, and division rates across the entire adaxial epidermis at early developmental stages, they found that cells in the proximal region already behave differently from cells in the distal region, even when both groups remain entirely green and apparently undifferentiated to the human eye.
At the earliest observable stage, when the petal primordium measures less than a millimeter in length and contains only a few hundred cells, proximal and distal cells already show statistically significant differences in their expansion rates and division patterns. The proximal cells tend to be smaller and divide more frequently. Distal cells expand to larger sizes before dividing. This differential growth creates subtle gradients in cell density and cell size that prefigure the future bullseye boundary position. Scientists describe this phenomenon as pre-patterning. The petal surface is effectively divided into distinct domains before any of the characteristics that make those domains visually different have appeared. Think of it like an architect's blueprint that marks where different rooms will eventually be built in a house, even though the actual walls, floors, and fixtures haven't been installed yet. The pre-patterning specifies future fates without immediately implementing them. This early specification involves genetic regulatory mechanisms that establish and maintain sharp boundaries between developmental domains. While the organ identity of the petal itself has already been determined by APETAL1 and APETAL3, the pre-patterning process subdivides this organ into functional regions that will later differentiate in distinct ways. As development proceeds, the differences between domains become more pronounced. By stage 1, when the petal has grown larger but still appears entirely green, cells in the proximal region have begun synthesizing the molecular machinery needed for anthocyanin production, though pigment concentrations remain too low to create visible color. The boundary between domains has sharpened, with cells on either side of an invisible line now following distinctly different developmental programs. Then, seemingly suddenly, but actually as the culmination of these earlier processes, color appears. Purple pigmentation spreads across the proximal region, filling in the domain that pre-patterning had specified stages earlier. The boundary becomes visible at last, but it was determined long before we could see it. This developmental timeline creates both opportunities and constraints for the plant. By establishing pattern boundaries early, the flower commits to specific proportions. But maintaining those proportions as the organ grows presents a challenge. From the stage when pre-patterning first becomes detectable to the stage when the flower opens, hibiscus petals increase in area by roughly 100-fold. How do you keep a boundary in the right relative position when all the tissue around it is expanding dramatically? Researchers built computational models to explore possible mechanisms. A one-dimensional model simulating growth along the proximal distal axis of the petal proved sufficient to capture the key dynamics. The model treats the petal as a line of cells that can expand at different rates and divide at different frequencies, depending on their position. By adjusting parameters controlling growth rates on either side of the boundary, the model can simulate how different growth strategies affect final pattern proportions. The simulations revealed 3 distinct strategies plants could use to maintain consistent bullseye proportions during growth. The first approach involves specifying the boundary position early, then ensuring that cells on both sides of the boundary grow at similar rates throughout subsequent development. If proximal and distal cells expand at the same pace, the boundary maintains its relative position even as absolute distances increase. The second strategy allows growth rates to differ between domains, but compensates by adjusting division rates. If distal cells expand faster, they divide less frequently, while proximal cells expand slower but divide more often, keeping the relative sizes of the two domains constant. The third mechanism involves modifying boundary position after initial pre-patterning by allowing differential growth and division in the regions immediately adjacent to the boundary, effectively pushing the boundary forward or backward during development. To test whether real plants actually use these strategies, researchers exploited natural variation within the Trianum species complex. Different hibiscus populations and closely related species produce bullseyes with different proportions.
Some have larger proximal regions extending nearly halfway up the petal, while others show smaller burgundy bases. By comparing cellular behavior across these variants, scientists could determine which mechanisms account for pattern differences. The analysis revealed that different populations appear to use different strategies. Some variants modify boundary position during the pre-patterning phase itself, specifying a different location from the outset. Once specified, they maintain that position through coordinated growth. Other variants establish the boundary at the standard position, but then modulate growth rates on one side or the other, shifting the final pattern proportions despite similar initial pre-patterning. These differential growth programs involve genes like rabbit ears and TCP5 that regulate the transition from cell proliferation to cell expansion, as well as factors like jagged that control growth polarity patterns, distinguishing petal development from leaf development. This flexibility in developmental mechanisms helps explain how pattern diversity can evolve. A mutation affecting just the initial specification of boundary position would create one type of variation. A mutation affecting growth rates in one domain would create another type. Both could produce viable flowers with functional patterns, just with different proportions. Natural selection or pollinator preferences could then favor certain proportions in certain environments, driving population divergence.
The functional relevance of these pattern variations remained uncertain until researchers tested whether pollinators actually respond differently to flowers with different bullseye proportions. Using buff-tailed bumblebees in controlled flight arena experiments, scientists presented the insects with artificial flowers featuring various bullseye sizes and asked whether bees could distinguish between them and whether they showed preferences. The bees proved remarkably sensitive to pattern proportions. They readily learned to associate specific bullseye sizes with sucrose rewards, successfully discriminating between flowers where the dark center covered 1/3 versus 1/2 of the petal area. This demonstrated that pattern differences evolution might produce fall well within the perceptual capabilities of important pollinators. Moreover, when given free choice without training, bees showed preferences for certain intermediate proportions over very small or very large bullseyes. The preferences weren't absolute. Individual bees varied in their choices, and learning could override initial biases. But the existence of any innate preferences suggests that pattern proportions could influence pollination success in ways that would create selection pressure for specific ratios. These findings from hibiscus raise broader questions applicable across flowering plants. If pattern boundaries are established through pre-patterning long before they become visible, what additional molecular signals beyond those we've identified specify and maintain these boundaries? How do cells on opposite sides of an invisible line come to follow different developmental fates when they originated from identical progenitors? What prevents boundaries from blurring as cells grow and divide?
The violet's nectar guides, the sunflower's ray pattern, and the complex regional divisions found in flowers like nigella all presumably involve similar pre-patterning mechanisms, where future domains are specified before differentiation makes them apparent. The petunia flower illustrates how patterns can vary not just across the petal surface, but along the proximal distal axis in ways that correlate with function. The limbs, the wide distal portions of the fused petals, feature smooth conical cells that attract pollinators visually. Moving toward the base into the tube region, cells become elongated and develop striations. Deeper in the tube, cells acquire central papillae. Each region likely serves different functions, perhaps in pollinator guidance, in providing grip for insects crawling through the tube, or in managing nectar presentation. These spatial patterns in cell differentiation require coordinate regulation across tissues. A cell's developmental fate must be determined not just by its intrinsic characteristics, but by its position relative to larger organizational frameworks. Understanding how such systems work requires bridging scales from molecules to cells to tissues. The pre-patterning mechanisms visible in hibiscus point toward a genetic architecture where transcription factors and signaling molecules create the invisible boundaries that later become manifest as the patterns we observe, leading naturally to the question of how this genetic machinery actually operates to transform early spatial specification into the differentiated tissues and vibrant patterns that characterize mature flowers.
Chapter 3. Genetic Architecture. Understanding how anthocyanins create color gradients or how cells arrange themselves into patterns requires looking beneath the chemistry and cell biology to the genetic instructions that orchestrate these processes. A flower doesn't spontaneously organize itself into sepals, petals, stamens, and carpels through random cellular decisions. Instead, a precisely regulated genetic program specifies which cells will become which organs, when those organs will form, and how they will differentiate into the diverse cell types and spatial patterns we observe. Without this genetic framework, the biochemical pathways that produce anthocyanins would have no information about where to activate, and cells would have no instructions about what shapes to adopt or what boundaries to respect. The breakthrough in understanding flower organ specification came with the formulation of the ABC model in the early 1990s. Through studying mutant plants with dramatic floral abnormalities, scientists discovered that flower organ identity is determined by the combinatorial activity of 3 classes of genes, arbitrarily labeled A, B, and C. The elegant simplicity of the system lies in how these 3 gene classes work together to specify 4 different organ types through their presence or absence in different positions. Consider what happens in a normal flower. In the outermost whorl, only A-class genes are active. Cells in this region respond to A gene activity alone by developing into sepals, the protective structures that enclose the developing bud. Moving inward to the second whorl, both A and B-class genes become active together. This combination of activities triggers a different developmental program, causing cells to become petals instead of sepals. The third whorl expresses B and C genes together, producing stamens, the male reproductive structures. Finally, the innermost whorl expresses only C genes, giving rise to carpels, the female reproductive organs. The logic becomes clearer when you examine actual mutants. A snapdragon plant missing functional B genes cannot produce either petals or stamens because both organ types require B gene activity. Instead, it develops sepals where petals should be, since the second whorl now has only A gene activity, and carpels where stamens should be, since the third whorl has only C activity. The resulting flower consists entirely of protective and reproductive structures arranged in the characteristic 4 whorls, but lacking the colorful petals that would normally attract pollinators. Such a plant can still self-pollinate if insects accidentally contact the exposed reproductive organs, but it has lost the elaborate signaling apparatus that would make pollination efficient. Similarly, mutations knocking out A gene function produce flowers where the first whorl develops carpels instead of sepals, and the second whorl develops stamens instead of petals. Walk into a garden and find such a mutant Arabidopsis plant, and you'll see a flower turned inside out, with reproductive organs occupying positions normally reserved for protective and attractive structures. The outer regions now express C alone, or C plus B, mimicking the gene activity patterns normally found in the flower's interior. These reciprocal transformations revealed something profound about how the ABC system works. A and C genes don't just specify particular fates independently, they actively repress each other. Where A is active, it prevents C from being expressed, and vice versa. This mutual antagonism creates sharp boundaries between domains, ensuring that cells commit to one fate or another rather than adopting intermediate states. A cell cannot become half sepal and half carpel, it must choose. The molecular identity of these ABC genes turned out to be equally illuminating. Nearly all of them belong to a family of transcription factors called Madsbox genes, named for the founding members identified in diverse organisms: MCM1, Agamis, Deficiens, and SRF. Madsbox genes encode proteins that bind to specific DNA sequences near other genes, regulating whether those target genes get transcribed into RNA and ultimately translated into proteins. In flowering plants, Madsbox genes underwent extensive duplication and diversification, creating large gene families where different members acquired specialized roles. The A-function genes include APTALA1, often abbreviated AP1. The B-function genes include APTALA3 and pistillata, abbreviated AP3 and PI. The C-function gene in Arabidopsis is called Agamis, or AG. The name reflects the phenotype. Plants lacking this gene produce flowers with petals and sepals but no stamens or carpels, rendering them sterile despite their abundant decorative organs. These Madsbox proteins don't work alone. They form multi-protein complexes, with different combinations creating the molecular machinery needed to activate the hundreds of downstream genes that actually build sepals, petals, stamens, or carpels. AP3 and PI proteins physically associate with each other, forming pairs that bind DNA more effectively than either protein alone. These pairs then interact with other Madsbox proteins like AP1 or AG, assembling into higher-order complexes specific to particular whorls. The formation of these tetrameric complexes, sometimes called the quartet model, allows for precise combinatorial control, where the same proteins participating in different complexes can activate entirely different sets of downstream genes. Later refinements expanded the ABC model by identifying additional gene classes. E genes, including members of the SEPALF family, are required across all floral whorls for any organs to develop properly. The E-function proteins act as essential partners for the ABC proteins in their multi-protein complexes. Without functional E genes, flowers fail to produce any recognizable organs, developing instead into structures resembling leaves. In one memorable mutant phenotype, what should have been a colorful petunia flower emerged as a whorl of green leaf-like structures, as though the plant had forgotten it was supposed to make a flower at all. D genes specify ovule identity within the carpel, controlling the development of reproductive structures where seeds will form. Together, these 5 gene classes create a hierarchical genetic framework, where organ identity in each whorl emerges from specific combinations of Madsbox protein complexes. The universality of this genetic framework across flowering plants proved remarkable. When scientists examined distantly related species like snapdragons, petunias, and even monocots like maize, they found the same basic logic operating. The specific genes might have different names, and their exact expression patterns could vary, but the principle of combinatorial Madsbox gene activity specifying organ identity held consistent. This conservation suggests that the ABCDE model represents an ancient genetic innovation that arose once early in angiosperm evolution and was then retained across hundreds of millions of years as flowering plants diversified into hundreds of thousands of species. However, knowing that AP3 and PI together specify petal identity still leaves enormous questions unanswered. These genes define that cells in the second whorl will become petal cells rather than some other organ type. But being a petal cell encompasses an extraordinary range of possible characteristics. A petunia petal cell looks nothing like a snapdragon petal cell or a hibiscus petal cell. Even within a single petal, different regions develop distinct cell shapes, cuticle structures, and pigmentation patterns. The ABC model explains organ-level identity, but doesn't directly account for the fine-grained differentiation that creates pattern and function within each organ. This is where downstream regulatory genes operating beneath the Madsbox layer become critical.
One key aspect of petal development involves controlling the transition from cell proliferation to cell expansion. Early in petal development, cells divide frequently, increasing cell number and establishing the fundamental size and organization of the primordium. Later, cell division slows or stops entirely, and growth continues through cell expansion alone. Individual cells can increase dramatically in volume during this expansion phase, sometimes becoming 50 or 100 times larger than they were when they stopped dividing. The timing of this transition profoundly affects final petal size and shape. In Arabidopsis, two transcription factors called rabbit ears and TCP5 play antagonistic roles regulating this transition. Rabbit ears, abbreviated RBE, promotes cell proliferation and delays the onset of expansion. TCP5 has the opposite effect, promoting expansion and suppressing continued division. The balance between these factors determines when cells in different petal regions stop dividing and start expanding. Research revealed that RBE doesn't just compete with TCP5 for control of shared target genes. Instead, RBE actively represses TCP5 expression through an epigenetic mechanism, recruiting proteins that modify chromatin structure around the TCP5 gene and making the DNA less accessible to the transcriptional machinery. By silencing TCP5 in regions where proliferation should continue, RBE ensures that the transition to expansion occurs only at the appropriate developmental stage. Mutations affecting RBE or TCP5 produce petals with altered proportions, sometimes dramatically smaller or larger than wild type, sometimes with irregular shapes resulting from uncoordinated transitions between growth phases. In severe cases, petals become so small they barely protrude beyond the sepals, or so large they dwarf the rest of the flower. These phenotypes demonstrate how genes regulating growth dynamics operate downstream of the organ identity genes to sculpt specific morphologies.
Beyond controlling when cells stop dividing, genetic programs must also specify regional differences in cellular differentiation within petals. The pattern boundaries established early in development, as explored in the previous chapter, must be maintained and elaborated by genes that respond to positional information. Different regions feature distinct cell types: conical cells that enhance color perception, flat pavement cells creating different optical properties, secretory cells forming nectaries, or trichomes providing specialized surface structures. The control of cell shape itself involves specific gene families. Mixed-alike MYB transcription factors play crucial roles in promoting the development of conical epidermal cells across much of the petal surface. When mixed genes are mutated or artificially suppressed, cells that would normally develop prominent conical shapes instead form flat surfaces, dramatically altering the petal's optical properties and texture. The mechanism involves mixed proteins activating genes that control cell wall composition and the machinery that generates the outward bulging of the cell surface. Pigmentation patterns within petals depend on additional layers of transcriptional regulation beyond the biosynthetic enzymes that produce anthocyanins. MYB and BHLH transcription factors work together to control where and when pigment production occurs. These regulatory proteins bind to the promoter regions of anthocyanin biosynthetic genes, activating them only in specific cells or at specific developmental stages. Different MYB proteins can activate pigment production in different spatial patterns, creating the spots, stripes, bullseyes, and gradients visible across diverse flower species. In some flowers, a particular MYB gene might be expressed only in proximal petal regions, creating a colored base while leaving distal regions white. In others, the same MYB family member might create radial stripes or spotted patterns, depending on its exact expression pattern and interactions with other regulatory factors. The boundaries between pigmented and unpigmented regions often involve genes like those from the cup-shaped cotyledon family, which help establish and maintain sharp domain boundaries during development.
These boundary genes prevent the diffusion of signals between adjacent regions, ensuring that pattern elements remain distinct rather than blurring together as the petal expands. MicroRNAs also participate in this regulatory architecture. Small RNA molecules like MIR164 and MIR172 fine-tune gene expression by binding to messenger RNAs and either degrading them or preventing their translation into proteins. These microRNAs can create sharp expression boundaries or temporal windows where certain developmental programs operate, adding another layer of control to the genetic networks establishing petal form and pattern.
The conservation of MADS-box genes and downstream effectors across flowering plants, combined with flexibility in how these genes are regulated and combined, helps explain both the unity and diversity of floral forms. Every flowering plant uses MADS-box genes to specify floral organ identity, but the specific patterns of where and when these genes are expressed can vary, as can the downstream genes they activate. Studies comparing AP3 genes across different plant families illustrated this principle. When researchers examined AP3 orthologs in snapdragons, petunias, grasses, and orchids, they found that the genes maintain recognizable sequence similarity and still participate in petal or petal-like organ specification. But the specific target genes that AP3 regulates differ between species, and the spatial patterns of AP3 expression within the flower have been modified through evolutionary time. In grasses, which have highly reduced flowers lacking showy petals, the B-class genes persist but function differently, helping specify the loticules, small scale-like structures that force the flower open during pollination. These observations point toward a crucial insight about floral evolution. Modifications to when, where, and how strongly genes are expressed, rather than changes to the genes themselves, become powerful drivers of morphological diversity. A mutation in an enhancer element controlling when a gene like TCP5 turns on could shift petal size without disrupting the gene's fundamental function. A change in the promoter region of a mixed gene could alter which cells develop conical shapes, creating new optical properties. Such regulatory changes can spread through populations rapidly if they provide pollination advantages or help flowers adapt to new environmental conditions. The accumulation of regulatory mutations over evolutionary time allows closely related species to develop dramatically different floral forms while retaining the same core genetic toolkit. The morning glory's broad funnel-shaped corolla, the snapdragon's bilaterally symmetric tube, and the columbine's elaborate spurred petals all develop using variations on the same basic MADS-box framework and the same families of downstream regulatory genes. The differences emerge from how those genes are wired together into regulatory networks, when they activate, where their expression domains overlap, and how strongly they function in different cellular contexts. This flexibility in genetic regulation connects directly to the evolutionary pressures flowers face from their pollinators. When a pollinator preference shifts, perhaps favoring flowers with deeper tubes or different color patterns, the genetic variation in regulatory elements provides the raw material for evolutionary response. Populations experiencing different pollinator pressures can diverge rapidly in floral characteristics because regulatory mutations affecting the spatial or temporal deployment of existing genes require fewer evolutionary steps than creating entirely new genes with novel functions. The molecular machinery we've explored here, from the MADS-box proteins specifying organ identity, to the transcription factors controlling cell division and expansion, to the regulators establishing pigmentation boundaries, all operate within the developmental framework that transforms a tiny floral meristem into a mature flower. Understanding this genetic architecture illuminates how the visible diversity of petal forms emerges from variations in gene expression and regulation, providing the mechanistic foundation for the co-evolutionary dance between flowers and their animal partners that continues to shape the botanical world around us. Chapter 4. The Pollinator's Perspective. When Charles Darwin examined orchid flowers under his microscope, he found structures so precisely matched to their pollinators that they seemed almost engineered.
One Madagascar orchid possessed a nectar spur extending nearly 30 centimeters, an absurd length that prompted Darwin to predict the existence of a moth with a tongue equally long. Colleagues dismissed this as speculation, but decades after Darwin's death, scientists discovered exactly such a moth, its proboscis perfectly dimensioned to reach the nectar at the spur's base while inadvertently collecting pollen on its head. This famous example illustrates a fundamental principle. Flowers and their pollinators haven't merely influenced each other's evolution, they've driven each other's diversification through millions of years of reciprocal adaptation. Coevolution describes this process, where two or more species reciprocally affect each other's evolution. When flowers with deeper nectaries attract more specialized pollinators, those flowers gain advantages in reliable pollen transfer. The pollinators gain feeding advantages from exclusive access to resources. The cycle continues, with each species tracking evolutionary changes in the other, sometimes leading to extraordinary specializations. This dynamic has produced what botanists call pollination syndromes, recognizable suites of floral characteristics associated with particular types of pollinators. The concept emerged from observing that flowers visited primarily by bees tend to share certain features, while flowers pollinated mainly by hummingbirds converge on different traits. These patterns aren't absolute rules. Many flowers attract multiple pollinator types, and some pollinators visit diverse flowers. But the correlations prove strong enough to predict a flower's likely pollinator from examining its morphology, color, scent, and nectar properties. Bee-pollinated flowers typically present bowl-shaped or tubular forms, providing landing platforms. Their petals display blues, purples, yellows, or whites, colors conspicuous to bees whose vision extends into ultraviolet but doesn't detect pure red as vividly as humans do. Many produce sweet fragrances and offer both nectar and pollen as rewards. The texture created by conical epidermal cells becomes critically important here. When bees land on petals tilted at steep angles or swaying in wind, these microscopic structures provide purchase, preventing the insect from sliding off before pollen transfer occurs. Research has demonstrated that bees visiting flowers with flat epidermal cells struggle significantly more on angled or moving surfaces, particularly in flowers like snapdragons, where the bilateral flower creates a closed tube positioned vertically or at sharp angles. The snapdragon's mechanism depends entirely on correct positioning. The bee's wheat triggers the flower to open, allowing access to nectar deep inside. As the bee pushes into the tube, its back brushes against stamens positioned to deposit pollen precisely where it will contact the stigma of the next flower visited. Without adequate grip, the bee cannot maintain proper orientation and pollination fails.
Some bees have evolved specialized behaviors matching particular flower forms. Long-tongued bumblebees access nectar in deep tubular flowers that exclude shorter-tongued species. Certain bees have developed buzz pollination, vibrating their flight muscles at specific frequencies that cause pollen to shake loose from flowers that hold it in tube-shaped anthers as tomatoes and blueberries do. The flowers benefit by ensuring only these specialized bees, which visit many flowers of the same species in succession, accomplish pollination. Bird-pollinated flowers diverge sharply from this bee syndrome. Hummingbirds and other nectar-feeding birds possess excellent color vision but relatively poor senses of smell. Flowers targeting these pollinators accordingly produce little or no scent while displaying brilliant reds, oranges, and other colors birds perceive vividly. The tubular shape appears again, but typically longer and more robust than bee flowers, matching the dimensions of avian bills and tongues. Nectar volumes are substantially higher because birds require more calories than insects. In New Zealand, native plants like kohai and flax evolved specifically for pollination by nectar-feeding birds. The flowers are large, sturdy enough to support birds perching or hovering while feeding and produce copious nectar. The kohai's flowers hang in clusters, and their curved tubular shape matches the curved beaks of the honey-eaters and other birds that pollinate them. When European honeybees were introduced to New Zealand, they could access some native flowers but proved less effective pollinators than the native birds for many species, lacking both the appropriate body size to contact reproductive structures properly and the feeding behavior that ensures pollen transfer.
The tree fuchsia, native to New Zealand, demonstrates how flowers can signal their pollination status to birds. When the flowers first open and need pollination, they appear green, relatively inconspicuous against the foliage. After successful pollination, the petals turn bright red, becoming highly visible. This color change tells birds which flowers offer fresh nectar rewards and which have already been serviced, directing pollinator attention toward unpollinated flowers and preventing wasted visits. Moth-pollinated flowers open a different niche entirely, operating primarily at night when bees and birds are inactive. These flowers tend toward white or pale colors that remain visible in low light. They release powerful fragrances, often sweet but sometimes complex or even slightly fetid, that carry through still evening air and attract moths from considerable distances. The nectar spurs or tubes are often extremely long, accessible only to moths with correspondingly long proboscises. Japanese honeysuckle illustrates this syndrome perfectly, its flowers opening at dusk and releasing intense fragrance to attract nocturnal moths that prove more efficient pollinators than the diurnal bees that occasionally visit during daylight.
Some flowers have taken night pollination to an extreme by specializing on bats. Bat-pollinated flowers are typically large, sturdy enough to withstand the impact of flying mammals, and often positioned on or near the main trunk of trees where bats can approach easily. They open at night, produce strong fruity or fermented odors attractive to bats, and offer abundant nectar or pollen. The dactylenthus of New Zealand represents an unusual case, a parasitic plant living entirely underground whose flowers emerge at the soil surface to attract short-tailed bats. Unable to photosynthesize, lacking colorful petals, the plant instead invests in powerful scent and rich nectar to lure the bats that provide its only means of reproduction. Wind pollination represents an entirely different strategy that has evolved independently in numerous plant lineages. Wind-pollinated flowers abandon the expensive structures needed to attract and reward animals. They produce no showy petals, no fragrances, minimal or absent nectar. Instead, they manufacture prodigious quantities of lightweight, smooth pollen that can drift on air currents. The anthers often dangle on long filaments outside the flower where wind can catch the pollen efficiently. Stigmas become feathery or branched, maximizing surface area to intercept wind-borne pollen. Grasses epitomize this syndrome, producing inconspicuous flowers that release clouds of pollen during breezy conditions. The transition from animal pollination to wind pollination apparently occurs relatively easily in evolutionary time, as evidenced by its repeated independent evolution. But the reverse transition seems much rarer, perhaps because regaining the complex suite of traits needed for animal pollination proves more difficult than losing them. This asymmetry has consequences for understanding floral diversity. Animal pollination creates opportunities for specialization and speciation as flowers and pollinators co-evolve. Wind pollination, while metabolically cheaper in some respects, offers fewer avenues for diversification. Between these extremes lie flowers with less specialized pollination systems. Many flowers attract a variety of visitors, from bees to flies to beetles. Bowl-shaped flowers like buttercups and wild roses present easily accessible pollen and sometimes nectar to almost any insect that lands. This generalist strategy sacrifices the efficiency of specialized pollination for the security of having multiple potential pollinators. If one pollinator species declines, others can substitute. Carrion flowers reveal how extreme specialization can become. These flowers mimic rotting flesh, producing colors ranging from dark maroon to purplish brown and emitting odors resembling decay. Flies and beetles attracted to carrion discover the flowers instead, and while they receive no nutritional reward, they often lay eggs on the flowers, mistaking them for suitable sites for their larvae. As they explore the flower, they accomplish pollination. The strategy works despite providing no benefit to the pollinator, exploiting the insect's sensory systems and behavioral programming. The strategy works despite providing no benefit to the pollinator, exploiting the insect's sensory systems and behavioral programming. Sexual deception takes this exploitation further.
Certain orchids produce flowers that mimic female insects so convincingly that males attempt to mate with them. The flowers replicate not just the visual appearance, but also the pheromone profiles of female bees or wasps. When a male insect tries to copulate with the flower, specialized structures attach pollen to specific parts of its body. The frustrated male eventually leaves and may attempt to mate with another flower of the same species, transferring pollen in the process. These various pollination syndromes create selection pressures that can drive populations of the same species to diverge when they occur in different environments with different pollinator communities. If one population experiences primarily bee pollination while another encounters mainly hummingbird pollination, the two populations face different selective pressures on flower color, shape, scent, and nectar properties. Over time, the populations may diverge so substantially that they become reproductively isolated, even if they could physically interbreed. This process, called pollinator-driven speciation, has likely contributed significantly to the extraordinary diversity of flowering plants.
The petunia species complex illustrates this process. Different petunia species and populations show remarkable variation in flower color, tube length, and scent profiles. Some species produce white flowers with long tubes and strong evening fragrance, attracting hawk moths. Others have shorter tubes and purple or red flowers visited by bees or hummingbirds. By comparing these species and conducting experiments where researchers manipulate floral traits or swap pollinators, scientists have shown that these differences directly affect which pollinators visit and how effectively pollination proceeds. When a flower evolves in tight association with a particular pollinator species, that flower becomes dependent on its partner's survival. If the pollinator population crashes due to disease, habitat loss, pesticide exposure, or climate change, the plant may lose its primary or only means of reproduction. Conversely, specialized pollinators, depending on particular flowers for food, face extinction if those flowers disappear. This mutual dependence means that biodiversity loss cascades through ecosystems in unexpected ways. Botanists have documented cases where rare plants persist in remnant populations but produce no seeds because their specialized pollinators have already disappeared from those areas. The plants bloom faithfully each year, but the flowers wither unpollinated, and the population slowly ages without recruitment of new individuals.
Climate change threatens these relationships through multiple pathways. Temperature shifts can alter the timing of flowering or pollinator emergence. If a plant begins blooming 2 weeks earlier in response to warmer springs, but its pollinator doesn't shift its life cycle timing correspondingly, the two species may miss each other entirely. Even if timing remains synchronized, geographic ranges may shift differently. A plant might track suitable temperature zones northward, while its pollinator tracks different environmental variables, leading to spatial separation. Documentation of such disruption is accumulating. Many flowering plant species in temperate zones are blooming progressively earlier, sometimes by days or weeks compared to historical records. Pollinator emergence times have also shifted, but not always at the same rates or in the same directions. Some plant pollinator pairs have maintained synchrony, while others have not, creating winners and losers in the reshuffling of ecological communities. Nectar itself represents an evolutionary battleground, where plant and pollinator interests sometimes align and sometimes conflict. From the plant's perspective, nectar is expensive to produce, requiring substantial energy investment. The ideal nectar reward would be just enough to attract pollinators reliably and keep them visiting multiple flowers of the same species without providing excess that wastes resources. From the pollinator's perspective, more nectar means more food with less foraging effort. The optimal nectar quantity and quality represent a compromise shaped by both the plant's physiology and the pollinator community it faces.
Nectar composition varies considerably across species, with different ratios of sugars, different amino acid profiles, and sometimes secondary compounds that affect pollinator behavior. Some plants add caffeine to their nectar in low concentrations that enhance pollinator memory and fidelity to that flower species without deterring visits.
Others include compounds that may discourage certain visitors while being tolerated or even preferred by others, potentially filtering which pollinators service the flower. These chemical manipulations reveal how sophisticated the evolutionary arms race between plants and pollinators can become, operating not just through morphology and color, but through subtle biochemical signaling. The position of nectaries within flowers creates another layer of control. Flowers visited by long-tongued pollinators often place nectar deep in tubes or spurs, where only those pollinators can reach. This exclusion strategy ensures that the pollinator touching the reproductive structures is likely one that can effectively transfer pollen. Nectaries positioned differently might allow a wider range of visitors, promoting generalist pollination. The balance each plant strikes between specialization and generalization depends on the reliability and diversity of the pollinator community it experiences, creating a tapestry of interactions where the morphological matching between flower and pollinator that evolved over millennia now faces unprecedented disruptions as human activities reshape the distribution and abundance of both flowering plants and their animal partners across the planet.
The next time you pause to admire a flower, remember that you're witnessing the culmination of an extraordinary convergence of sciences. From the quantum mechanics of electron excitation in anthocyanin molecules to the nanoscale architecture that bends light into iridescent blues, from the genetic programs that establish invisible boundaries in developing buds, to the millions of years of coevolution between flowers and their animal partners, every petal represents a masterpiece of natural engineering. These systems, refined over evolutionary time, now face unprecedented challenges as human activities disrupt the delicate timing and spatial relationships between plants and pollinators. Understanding the science behind petals isn't merely an academic exercise. It reveals the intricate dependencies woven through ecosystems and highlights what's at stake as we reshape our planet. Perhaps the greatest lesson these petals teach us is that beauty in nature emerges not from simplicity, but from layers of complexity we're only beginning to comprehend. Thank you for exploring the science of plant petals with us.